Today we can discuss about the chromatography. Chromatography is the versatile technique. What is meant by chromatography? Chromatography in before going to the discuss about the chromatography, how the chromatography introduced in the global, that is Michael Svet, a Russian scientist, 1972 to 1919, botanist, in 1906. Svet used the chromatography to separate plant pigments. Plant pigments. Michael Svet is a Russian scientist. Actually, he is a botanist, but he discovered a lot of new techniques by using by that is called as the chromatography. In 1906, the plant pigments, small, tiny pigments are separated by using this technique. He called the new technique that is called the chromatography because the result of analysis after completion of this technique, the results was came that results was analysis was written in color along the length of adsorbent column, that is called written in color. Color means the chroma means color, graphy means right. Whatever the pigments present in which color is present, just like that, you have to write it in the graphene. That's why you have to call it as the chromatography. In other words, we can say that is called chromatography is an application in every branch of physical and biological sciences. What is the use of chromatography in the global and entire world? Because the chromatography has an application in every branch of the physical and biological sciences. In the biological sciences, we like just like our botany, zoology, and biotechnology, in the whatever it may be in the bio, that is called the living organic sciences, that is very useful, and physical sciences just like chemistry and physics. And another one most important thing is for the chromatography, about 20 Nobel prizes, that is the miracle, awarded since 1937 alone for our work in the chromatography played in the vital role. Because how the Nobel Prize gets, new inventions not get any Nobel Prize. That is the source of inventions. Source of inventions. Whenever the source of inventions is discovered and find out, then only got the Nobel Prizes. In 1937, from 19, since 1937, 20 Nobel Prizes awarded from this chromatography, that is a vital role in the sciences, life sciences, and as well as the physical sciences. This is the very wonder in the world in the you get the Nobel Prizes. Suppose how the in the sweat scientist in previously told that botanist sweat scientist, what he done in experiment for the pigments, plant pigments for the experiment. In this experiment, the leaf extract, suppose you take the leaf extract sample in petroleum ether, that is called the solvent, was dissolved and allowed to pass through the column. In the column, what contains the column? In the column contains the calcium carbide. Simply you have to take the calcium carbide, that is called CaCO3. That pure ether was continued to flow through the column. As a result, continued flow of the through the column as the respective manner in which the various chlorophyll pigments are the very tiny particles that is separated by in a series of differently colored and easily distinguished zones. Easily distinguished means that you have to separate the color zones, light differentiation or very tiny differentiation, whatever it may the color is present is automatically we have to distinguish zones. This experiment is done by the sweat scientist botany. What he done? He leaf extract. Simply you have to take the leaf extract. Petroleum ether, that is called solvent, pass through the column. What is the in the column present in the calcium carbonate? Pure ether was continued to flow through the column, as a result in which the various chlorophyll pigments were separated into series of different colored. Uh, yeah. What is the definition in that particular area of the definition in for the chromatography? In this method, the separating in the chromatography, you have to separate the mixture of the components in individual components through the equilibrium distribution between the two phases. This most important aspect is the keyword, that is equilibrium distribution. Whenever the components is present, what are the components? These are the individual components. Individual components are present in the two phases, present in the mixer components, no problem. But we have to separate by distribution in equilibrium between the two phases. What is the two phases? What is it? Is any two phases. That is, it is analytical technique for resolution of the solute. The chromatography is the analytical technique. We have that analytical procedure. We have to adapting the analytical procedure. That is called the That's why it is called as the analytical technique in the resolution of the solute. Now, the resolution means the solute can be separated, easily separated. How to separate it? This is called very small quantity of the solute is also separated. That is called the resolution of the solute, in which the separation is made by differential migration. Differential migration in porous medium, and migration is called flow of solvent. Migration of the porous medium and migration is caused by, that is called flow of solvent. Why? How to migration? That migration is caused by the flow of solvent. That is the technique. Uh, Now we have to discuss about the what is the basic principles of the chromatography. Basic principle of the chromatography can be defined as a resolution in the multi-component mixer. Multi-component mixer means, so your mixer contains the so many compounds, so many solutes are present, some x to z or 1 to 10, whatever it may be there, the resolution that is completely resolved, completely separated in several hundred cases. Distribution, that is called the distribution between the, already we have to mention about the two phases. One is called the stationary phase, and is the one is the, and another one is the mobile phase. What are the two phases distribution? Stationary phase, that is the constant phase. Constant phase means that is not any moving from one place to another, that is called stationary phase, constant. And the another one is the mobile phase. Mobile phase means that is called one place to the travel to the another place, that is called mobile place. That is the basic principle. The principal mechanism, how to separate, so the separation of the difference, separate depends upon the differential solubility or adsorptivity of the mixer components with respect to the two phases involved. Two phases involved, that is a differential solubility. Some compounds are solubilities are slightly soluble, some compounds are easily soluble, some compounds are saturated solution. Just like that, the adsorptivity of the mixer of the components with respect to two phases involved. That is, these components must exhibit whatever it may be different partition coefficients. That is the mention about keyword in the chromatography basic principle. That is the different partition coefficients. The stationary phase may be solid or liquid, and the mobile phase is either in the liquid or gas. You have to mention about, please remember that the stationary phase may be liquid or solid, whatever it may be. The mobile phase is either liquid or gas. That is the most important for the two phases. And another pure solvent, the mixture of the solvents of a gas is allowed to move slowly other stationary phase, that is called mobile phase. Moving phase is called as the mobile phase. Mobile phase means one travel to the another place from left to right or bottom to top to bottom, bottom to up, that is called mobile phase. This is also called as the developing of the solutes in the resolution. That's why that is called the developer. That is also another words we can say, that is called eluent. Eluent means that is called the developing of the your solutes present in the mixer of the components. That is the basic principle of the components. Now we can discuss about the chromatography technique. I already mentioned about that, separating the mixer into their components. In What is the chromatography? Analyze, identification, purify, and or quantify the mixture of the components. So we have to take the mixture of the components, that is called the, you see that components yellow balls. It is called another yellow balls. It is called three yellow balls are there, circles. In this yellow contains the, and the red balls also present in that. Four red balls is also present. In addition to that, we have to addition of the blue. rectangular shapes of the components, three components present, and another one is the two black circles also present. How many mixtures of the components? There are the four different components are present, four different um, solutes are present in the mixer. We have to separate by using the chromatography. We have to the components resolved. This is the yellow, this is the red, this is the blue, and this is the black. And we can separate the all the components of this chromatography. We can use the and after collecting of all these four components, we have to analyze that component compared to the other, and identify what is the red, what is the yellow, what is the blue content, what is the black content. We have to identify, and how much quantity is present. Is there any impurities present in the yellow balls? Is there any present in the red balls? Is there any present in the blue? Is there any impurities present in the, just like the in black? That's why you have to calculate the purify and quantity. How much quantity is present? How much quantity is also present in the chromatography technique? This is the simple. We have to what is meant by chromatography. Mixer we have to take and separate by using the chromatography technique. All these components are separated, and then we have to analyze, identify, purify, and quantify. What is the use of chromatography? We have to scientists that examine the mixer. It is a components. There is a relation to one another. Identify the components. Determination of the identify. How to identify that mixer components based known components. That is called known components. Purifying. Separate the components in order to isolate one of the interest for further study. Further study we have to use that to purify or impurities are present. We have to that is most useful for the further study. In the quantify, how much quantity of the amount of mixer and are the components present in that sample. These are the all parameters we have to use this in the chromatography. In the real time, what is the life examples of the chromatography? What is the general uses of the chromatography? You have to use the pharmaceutical company. Determines the amount of each found in the new product. Determine the amount of each chemical found in the new product. We have to take the chemical reaction from 1 to 5 steps. From one step to the second step, you get the another product. That product is correct or not? What is the ingredients are present? What is the percentage of our target material is present in that particular sample? We have to analyze. We have to how much quantity is present in the particular first step. That is also in the pharmaceutical, and we have to go to the second step. That is called second to third step. We have to use that particular component. How much quantity is present in that present in the sample, and the second to third one, third to fourth, fourth to fifth. All these five reactions we have to completed the product. We have to analyze in the pharmaceutical company. That is the most useful, that is most advanced technique in the chromatography. We have to generally we have to take in the public scenario. We have to take the hospitals. We have to take these are any sick in the living system, and that is in the animal or whatever it may be the human beings. We have to detect the blood or alcohol levels in a patient's bloodstream. What are the contents in your bloodstreams? Is there any inorganic elements just like our potassium, sodium, huh? Just like our magnesium, selenium, whatever the elements are present in our bloodstream, you have to use this technique also, chromatography. In the law of enforcement, the compare that sample found at a crime scene to sample from suspectable. Is there any suspectable person? You have to collect that and compare the sample found. We have to initial and after we have to take that one sample, and that is also most useful in the enforcement. And is the most important thing is the environmental agency. Environmental agency means determine the level of pollutants. That is water supply. We have to take the municipal water supply and the bore water and the agriculture water supply, whatever it may be. Is there any water supply? We have to determine the how much level present in that particular water supply. We have to also use for the lot of use by using that chromatography. We have to determination. And the another one is the manufacturing plant. Is there any manufacturing plant? The purify the chemical needed make a product. That is the any manufacturing plant. We have to purification of the chemical needed to make the new product is also most useful. uh, this is called the we have already discussed about the detailed definition. Chromatography is the laboratory technique that separates the components with a mixer using the differential affinities of the components. Mobile medium means that is called phase. For stationary absorbing means stationary means that is only for the absorbing. Please remember that one, that is called the absorbing medium through which they pass. In the in the terminology, in the differential, showing the difference, distinctive affinity, natural attraction or force between things. Mobile medium, gas or liquid, carries the components mobile phase. Stationary medium, the part of that apparatus does not move anything in the stationary phase. Um, you have to take the illustration of the chromatography technique. We already mentioned about that. We already told that the chromatography, this is called mixer. This is the in the chromatography. This is the yellow stimulus. This is called mobile phase. Mobile phase in that particular chamber. In that, this is called mixer spot. It contains three yellow balls, three red balls, and three violet blue color rectangular, and two dots are present in the black. This is the chamber we have to separate. You have to take this is white this is called the stationary phase. This is called the mobile phase. Mobile phase means mobile phase contains that mobile phase. That mobile phase contains the actually you have to take the solvent from it moves from one place to the another place. All the components, all the whatever the present in our sample, that moves from bottom to up, from bottom to up by moving phase. That is called mobile phase. We have to separate by using this technique that is called the chromatography. You see the example, blue are three, these are present, and the black spots are present in that one, and then go to the red balls, and then go to the that is what you call yellow. These are all is separated by using the chromatography technique. That is called the stationary phase. This is called stationary phase. Yellow is the mobile phase. This is the yellow phase. Before going to experiment performance, after completion of the experiment, the mobile phase from starting this to this end, along with that is along with the all the four different solutes were present in the particular sample. This is the illustration. In the classification, that is the most important for the chromatography, classification of the chromatography. According to the mobile phase, there are the two types are present. According to the mobile phase, that is only classification mentioned about the mobile phase. One is the liquid chromatography. That is called the mobile phase is only for the liquid. That is called the liquid chromatography. Another one is the LLS. That is called as the liquid chromatography. LSC means liquid solid chromatography. Liquid solid chromatography means liquid is the chromatography, that is called mobile phase. Stationary phase is the solid, that is called LSC. And what is by LLS, that is called the liquid is the mobile phase, liquid is the stationary phase, that is called the LLS. And the another one is the gas chromatography. Gas mobile phase is a gas, that's why you have to take the GSC and GLS. Gas is mobile phase, and the solid is the stationary phase, and liquid is the stationary phase. That's why that is called as the GSC and GLS. These are the two types of classification of the chromatography. Another one is also classification, that is according to the packing of the stationary phase. Packing packing of the stationary phase, you have to the how to packing of that phase, that is called the very thin layer is present in that particular phase, that is called stationary phase. The stationary phase is thin layer, very thin layer, supported on a glass, plastic or aluminum plates, that is called thin layer chromatography. Paper chromatography, the stationary phase is a thin film of a liquid supported an inert support. Inert support means stationary phase, very thin film. Liquid is always inert support. That is called paper PC. That is called the thin layer chromatography, TLC. And another one is the most important, that is the column chromatography, that is also called as the CC. Column chromatography, stationary phase is a packed in a glass column, long, huge column, that is called along the big column by using the packer, that is called the column chromatography. And we have to take that this is the another classification of that material according to the force of separation. Separation is force, that is the random force. By using that force of separation, we have to classification is the 5. five types of the classification. One is the adsorption chromatography, and the second one is the partition chromatography. Third one is the ion exchange chromatography, that is called the gel. Another one is the third is the gel filtration chromatography. Fifth one is the affinity of the chromatography. These are the classification according to the force of separation. We have to classification separation means in the types of mobile phase. We have to take the gas liquid chromatography. A mobile phase is a gas. Stationary phase is the liquid. What is the method of fixing stationary phase? Adsorbed on porous solid held tube or adsorbed on the inner surface of the capillary tube. This is another one. Another one is the gas liquid. Gas liquid solid chromatography. Mobile phase is a gas. Stationary phase is a solid. Partition chromatography. Types of mobile phase is a liquid, and the types of stationary phase is the liquid. Adsorption, that is called liquid and solid. Paper chromatography, that is called the liquid, and the liquid is the stationary phase as well as the mobile phase. And the thin layer is the liquid phase, mobile phase, liquid or solid. Ionic exchange chromatography. Mobile phase is a liquid, and types of state phase is the solid. These are all the classification of the chromatography methods. It generally we have to take the adsorbents, that is called the stationary phase. Whatever the adsorbent, that is also called as the stationary phase. Adsorbent always, stationary phase always maybe adsorbents. What is the most important for the requirements of the good adsorbents? It should not be still suitable with the solution under the examination of the illusion. It is desirable to the colorless, always to the adsorbent may be colorless. It should be chemically inert with mixture of the under analysis. Always you have to take the adsorbent is the inert under the analysis. In the the particle size of the material plays the most important role in the chromatography. It is most widely used in the adsorbent, that is called alumina. The particle size of the alumina is the 50 to 200 pm, which allows the relatively. This is the adsorbent. This is called stationary phase. What are the good characteristics of the good adsorbents? Is there an adsorbent? Is it good or bad? Whatever it may, how to classification? There are some characteristics. Is there obey that characteristics? That is the good adsorbent. We have to call it as the good adsorbents. That is called large surface area. Environment is available of the polar sites. Reproducibility in the degree of the activation. One is the large surface area. Large surface area means we have to take the adsorbent. Whenever the using the chromatographic technique, the adsorbent, that is called stationary phase, is large. That is the good adsorbent, no doubt at all. And another one is the polar sites. We have to take the readsorbent in the stationary phase. Available of the polar sites is more and more. That is the most important good characteristics of the adsorbent. Another one is the reproducibility in degree of activation. Activation is the more common thing, but reproducibility is the most important for the good adsorbent. Now, so you have already mentioned about that. We have to already absorb mentioned that we have to take the classification of the adsorbent force. Classification of force according to the force operating in the adsorbate. Whenever we have to using the adsorbate, the force is applied fast, slow, whatever it may be. Force is very weak. Your applied force is very weak. Generally, we have to use the adsorbent like starch, sucrose. Sucrose are used for that weak. The in the weak force. Generally, force is intermediate. That is called the between the strong and weak. We have to apply that force. The calcium carbonate, calcium phosphate, calcium hydroxide, or magnesium hydroxide, whatever it may be, hydroxide, most suitable for the adsorbents. Suppose you have to take the force is very strong, then alumina, Al2O3, charcoal are used. Al2O3, the most common adsorbents, alumina and silica gel. You have to take the silica gel, Al2O3 and silica gel. These are the classification of the adsorbent, that is the stationary phase. Another one is the eluents. What is the mobile phase? Development of chromatogram. The chromatographic tape development after the changing of the column can be done by three methods. Another one is the frontal analysis, illusion analysis, displacement analysis, and displacement of the development of the analysis. Illusion technique. What is illusion technique? Illusion means that is nothing but a mobile phase. That's all. But eluents which may be pure solvent is passed. That is called 100% pure solvent. You take, that is mandatory. Suppose you have to take the less than 100% pure solvent, that is not suitable for the chromatographic technique. That's why you have to take the pure solvent, that is called 100%. Is passed through the column, which turns leads to the differential migrations. Migrations are the different. That is the 10% migration and the 50% migration and the 100% migration. Solutes in the mobile phase. If the flow rate is fixed, suppose you have to take the flow rates is fixed, separation depends upon the value of Kd. And another one is the eluting agents, solvent mobile phase. In the description we have to choosing the eluting agents. First one is the solvent should satisfy. Whenever you are performing the experiment, solvent always should satisfy the practical factors such as the viscosity, stability, and comparability of the detector solubility with respect to the sample. That is the most important characteristics of the solvent. And another one is the solvent always should provide the maximum resolution. That is a good solvent. You have to use, that is called eluting agent, good eluting agent. Solvent acts as a mobile phase. That is good mobile phase. Whenever the satisfied with all these things, that then only you have to call it as the solvent is a good solvent mobile phase. Solvent should provide the maximum resolution for the separation of the reasonable time. What is meant by reasonable time? That is called the little bit, not more than 5 minutes, not more than 10 minutes, not more than 15 minutes. Within the 15 minutes, within the 10 minutes, within the 5 minutes, we have to complete the experiment, and then performing the experiments, then we got the good result. That is the most important for the solvent. Is there any functions of the eluting agents? Is there any functions of the solvents? These are the required solution mixer can be separated. Effects process the development which various bands. Chromatography separated from each other, and the solvent used, that is called the eluent, to remove each band from the developed the chromatography. Is there any good eluent, good solvent? Must be capable. It is must be capable for dissolving the reality reality of the various components of the column. Components must be detected, various columns, various components present in that particular column. In the another one is the good important thing is the solvent with a high value of eluent strength, very high value, which will absorb the spaces more rapidly than within the value. Another most important, how to calculate that components present in that general chromatography. We have to take the RF value, retention factor, retardation factor. RF value is the symbol of the capital R. In the suffix we have to F, that is called the factor, retention factor. The value is the symbol for the retention factor. RF value denotes the ratio of the distance moved by the component from the and the origin by and the distance moved by the solvent from the origin. That's why it is represented mathematically represented as RF is equal to distance moved by the component distance moved by the solvent front. RF, most important thing you have to remember that what is the values of RF? RF values always less than 1. Most important thing. Is there any value your calculated RF value greater than 1? That is quite wrong. Absolutely wrong. That's why we have to take the RF value, because the distance moved by the component is always high. Distance moved by solvent is always high, distance by compound is always less. The factor in mathematically, that is the point something coming, that is called 0.98, 0.89, 0.78. Whenever the RF value is calculated less than 1, that is the most important thing for the RF values. Then only you have to calculate the in the chromatographic technique. Is there any effect of the RF value? Is there any factors affecting all the sure, we have to. So many factors are for the affecting the RF value. One is the value depends. That value depends upon the nature of the mixer. Mixer is the different. then only automatically your RFL is also different. That is, the mixture is the nature. What is the nature? Solvent or solute, just like that. Different types of particle size. That is also for the RFL is the value is the variation. Another one is the nature of solvent. Solvent is also the most important thing. Solvent. Whatever the solvent is present, nature is the most important for the factors affecting the R value, and the another one is the most important for the medium is used for the separation. Whenever the medium is also most important for the separation, medium is not suitable for that resolving of the components, then automatically R value is maybe vary. That's why in the similar manner we have to the quality of the filter paper used. Is there any paper is for the quality, quantity of the paper? Quality of the paper is used for the factors affecting the RF value. Suppose you did not use any quality of the paper, automatically RF value may be changed. And then another one is the most important thing is the temperature. Temperature. We have to performing the experiments in the room temperature only. This is below 30 degrees centigrade. Then only got the good values. After experiment is the always same experiments or repetition or in another manner we have to calculate the RF value in the same temperature. Temperature is also most important. At what temperature you have to mention that you are performing that experiment by using the technique of the chromatography. Temperature is mentioned about 25 degrees centigrade or about 27 degrees centigrade, not more than 10 degrees centigrade. And another one is the size of the car, that is called container. Container is also most important equipment carried out. Is suitable for the container size, that is also RF value is most important factors affecting. And RF value, is there any identification for the RF value? Is there any helpful for the identification of the component? Yes, that is most important for that RF value used to utilization of that RF value. We have to identifying that component. Same adsorbent, same solvent, performed the same temperature. That is the constant RF value. Suppose you have to take the RF value is different, definitely we have to the deviating of the all the parameters are the not same. That's why in the constant RF value provided same adsorbent, same solvent and same temperature. In knowing the RF value, comparing the standard values different from the analytic of the component. Analytic of the component. Measurement of the RF value, we have to take that which type of solvent used. The similar RFL do not indicate that components identical. For example, RFL is the amino acids of the, suppose you take the alanine, glycine, and another amino acid, tyrosine, in phenol, water system is the same because the in RFL is the 0.55 is the most important. It is the necessary to use the different systems for identifying of the different components. Different. This is the most important. This is the generally briefly already told that by using the chromatographic technique in this class.